Okay, so hope everybody's doing all right. Today we're going to talk about energy generation. That is how cells manipulate energy and I guess you can say store energy, see in the form of ATP. Of course, construction of a cell requires an enormous amount of energy. There's energy required for protein synthesis, nucleic acid synthesis, building larger macromolecules, construction of the cell, muscle movement. And we need an enormous amount of energy. Now, ultimately, energy, of course, comes from the sun. All the energy that's basically on Earth originated from sunlight. The energy stored in sunlight was used by, or is used by plants, chloroplasts, and chlorophyll in order to carry out photosynthesis, which we'll talk about next time. And then, using photosynthesis, plants build compounds like small sugars and larger sugars. And the way we get our energy is from those bonds. So all the energy basically is stored in bonds. And the energy is extracted from bonds, and then we use that energy. Now, the reason I'm talking about this now in mitochondria electron transport now, and the readings I have written down below here, these are the readings that focus on what we'll be talking about today, page 81 through 87, chapter 13, and so on. But the reason why I'm talking about energy today, although it is towards the back of the book, and like I say, I organize topics depending on how they make sense to me. I don't always follow precisely the book, chapter 1, chapter 2, chapter 3. I kind of put it together in a way that makes sense. But energy storage and energy manipulation is extremely important. And what I have found in the past is that if I don't put it at the very, very beginning of the class, like I am now, then I usually run out of time towards the end, and then I don't present it at all. And I think it's really important to look at this and get an appreciation of how mitochondria work and what electron transport is. So we're going to put it here. And next time we'll talk about a little bit about photosynthesis. But the bottom line is that the energy is stored in all energy that we use is basically stored in chemical bonds. So what I'm showing here basically is that all the energy that we use, the cellular energy, is ultimately derived from sugars, carbohydrates, fats, proteins, food that we eat. And the energy is stored, like I said, in the chemical bond. So if you take glucose, for example, C6H12O6, and you oxidize that in the presence of oxygen, you get combustion, you end up completely breaking down that molecule into carbon dioxide and water, and then the energy of all those bonds is released. And it actually is quite a bit of energy, and if you look, just take, for example, you take a piece of paper and burn it. The flame that's coming off of that paper is due to the oxidation of the bonds that's holding that paper together. If you light a match or you burn a piece of wood, I mean, there's an enormous amount of energy that's released. From the book, this basically is showing the direct burning of sugar. So if you take sugar in the presence of oxygen and you increase the energy, say you light it, then all that energy is released in that flame. So it says all free energy is released as heat, none is stored. Of course, we generate CO2 and water as the combustion products. Now, what we do, or what cells do, is a little bit different, because what we were trying to do is to capture that energy. So that's kind of illustrated here again, figure from the book, and that is we take sugar, or it could be fats, it could be lipids, it could be protein, other carbohydrates. We take that sugar in the presence of oxygen, we ignite it, and then what we do is we capture the energy in pieces. We we break down the sugar. Instead of one combustion product, we break down that sugar in steps, stepwise. And we capture the energy coming off of those steps, and we use it. And as it says, some of the energy is stored in the activated carriers, and of course at the end you get the oxygen, the CO2 and the water. But the point is that we capture the energy as it's being stepwise degraded, rather than just carrying out full combustion. So we come back to this figure, and of course, so I just want to back up a little bit and say that a lot of what I'm going to show are figures that I have drawn. Like I mentioned earlier, a couple days ago, I traditionally would write a lot of things on the board, and now that we're going to this online version of teaching, so I kind of had to draw on the board, so I converted that into PowerPoints and put it together with other things from the book. But in many cases, the figures from the book, they may not describe precisely what I want to describe. And so I make my own drawings that hopefully will express some of the things that I want to express a little bit more clearly. And the truth of the matter is that if I just use, a lot of people just use the PowerPoints from the book. They just show figure after figure after figure, and I don't think that's really useful. If that was the case, then you wouldn't need anybody to lecture to you about this. All you need to do is read the book and take a look at the pictures. But what I do is I try and interpret and kind of refine some of what's there and represent it in a way that makes sense to me and hopefully makes sense to you. So a lot of these figures that you'll see throughout the semester were drawn by me. Okay, in any case, so we take the bonds that are in glucose or any of the other food products, and we basically hydrolyze those bonds, energy is released, and we recapture the energy in bond form. So we convert the bond energy back into bond energy. And the energy is converted back into bonds of a very specific molecule called ATP. ATP is adenosine triphosphate. That's the structure of it on the bottom of this slide. And ATP is considered to be the universal energy carrier of the cell. So the energy that's held by ATP is really in those three phosphates that are on the left-hand side. And the issue with these phosphates is that they're highly negatively charged. You can see the O minus, O minus, O minus on the bottom, and also the oxygens at the top, which are double bonded, they're negatively charged. So it's in principle, it's hard to get negatively charged molecules together. They repel each other. The negative charges repel each other, and so in order to make these bonds, a lot of energy needs to go into the construction, and so they hold a lot of energy. So when those bonds are hydrolyzed, a lot of energy is released. Now ATP is used as pretty much the entire energy source of the cell. Sometimes GTP is used, but mainly it's ATP, and it's the bond energy in ATP. So you might ask the question, why do we need ATP if there's bond energy in the glucose? If there's bond energy in fats or proteins? Why can't we just use that bond energy directly? Why do we have to transfer it into another bond? Well, the idea is that, in fact, in principle, we could be able to extract the energy and use it from a sugar, directly from those bonds. But in order to do that, in order to make it useful, you need to have different biochemical mechanisms, different mechanisms that would use different energy sources. So if we were using, for protein synthesis, if we use sugar, the energy from sugar bonds, you need a whole biochemical series of reactions to do that. Or if you were synthesizing nucleic acids and they use a different energy source, you need a different series of mechanisms to get the energy. And so the way the cell is kind of consolidated its resources, that we use one energy source, which is basically ATP. And so all mechanisms that require energy then kind of use the same mechanism to extract that energy. We kind of consolidate it all in one. It's like all all cars, at least used to use gasoline. And now we use gasoline and ethanol and electricity, but it's using the same source for same mechanisms for all the different reactions that take place in the cell. So it's transferred back into ATP. Now the bond energy is released and it's recaptured by a process known as chemiosmotic coupling. Well, let's look at the very bottom. That kind of shows a little bit the outline. So the sugar, which is the bond energy, where all that energy is stored, that we ultimately want to extract. Then through a series of complicated chemical reactions, which I'm not going to talk about, I'll mention that in a second again, glycolysis, the TCA cycle, we end up moving electrons and protons into the electron transport chain. And so that bond energy gets converted into what's called potential energy. And that potential energy is then converted back into bond energy. So we go from bond, potential, back to bond in ATP. And that occurs via what's called chemiosmotic coupling, and that takes place in the mitochondria. And there's two pieces that I think are kind of most important. One is the electron transport. That's the production of a proton gradient, which we'll describe. And then there is a protein complex called ATP synthase, which synthesizes ATP. And we're going to go through some of these things in a little bit more detail. Now here's kind of a larger scheme showing at the top of food. You digest the food, you get simple sugars, amino acids. It gets fed into glycolysis, generates pyruvate, and it goes into the mitochondria, and you see acetyl coenzyme A there, and then there's citric acid cycle. Now, I'm not all that interested. I'm interested in it, but I'm not all that interested in presenting the biochemistry. We're not going to talk about glycolysis, and we're not going to talk about the citric acid cycle. There are a whole series of reactions that are quite complex that I don't really think are necessary for what I want to emphasize, because I want to really talk about the strategy that the cell uses or the mitochondria uses. And in any case, I think that the reactions that take place in glycolysis and the TCA cycle are more in the realm of biochemistry. So hopefully when you take a biochemistry course, or if you already have, you've had these things in enough detail. But the main point this slide here, and we'll look at a little bit more detail in a second, is that in the cytoplasm, that's the gray area, the glucose gets hydrolyzed. There's a little bit of ATP produced, a little bit of NADH, and you end up getting pyruvate. I want to look at pyruvate in a second. That's just basically at the next slide. But before I finish, let's just look at the very bottom. So it kind of gives that outline again. You go from food, and you end up in the presence of oxygen. You end up getting ATP and NADH, and of course the carbon dioxide and water. So that's the combustion, but we, instead of releasing it as energy, we get the ATP out. So here's a very skeletal diagram of the breakdown of glucose. Of course, this takes place in glycolysis. There's 10 reactions, which we're not going to be looking at. But the end result of glycolysis, this takes place in the cytoplasm, are 2 molecules of pyruvate. And you can see the 2 molecules on the right-hand side. In the process, 2 molecules of ATP are used to start the whole. So here's that illustration again from the text. So the cytoplasm represents the cell. The cytoplasm is the gray area. The mitochondria is the whiter area. And so through glycolysis, you end up with pyruvate. And And that pyruvate then is transported into the mitochondria. And then that pyruvate gets converted to acetyl coenzyme A, and I want you to take a look at that in a second, but that acetyl coenzyme A gets fed into the citric acid cycle, which again, we're not going to talk about the reactions, that's more for biochemistry. And then you end up with what it shows here is NADH feeding into electron transport, and we'll talk about that in a minute. But I want you to take a look at so here's an image of the structure of acetyl coenzyme A. I just want you to take a look at this. It's not a very complicated molecule. Looks a little bit like ATP, in fact. Look at the bottom. It's a nucleotide. If you look at the bottom, there's a sugar, and then there's a base attached to it. And then off of that carbon towards the bottom, there's two phosphates. So this is basically a nucleotide, an RNA nucleotide, similar again to ATP. That part of the structure, and then attached to that end phosphate is a longer organic molecule, and then eventually at the end on the left-hand side, you see the acetyl unit, which is added onto this coenzyme A. So this is acetyl coenzyme A, and what's left of the sugar is the CH3 double bond O and the bond to the sulfur. So all of the energy that was in the bonds of the sugar is maintained, and it's held by the bonds in that acetyl unit at the very end. That part of the molecule came from the sugar, and there were two molecules of acetyl coenzyme A that are generated, and so that the bond energy is now held there. What was in the sugar is now held here, and this kind of, again, just shows what I want to illustrate. So, of course, the goal is ATP synthesis in the mitochondria, and that's kind of where we're going. So you have the polysaccharides, lipids, and the carbohydrates, end up with glucose that gets fed into glycolysis in the cytoplasm. You end up with pyruvate, and if you look at pyruvate, what it is in essence, just the sugar cut in half. The glucose is C6. If you look at the pyruvate, there's 3 carbons. So half the glucose is now, except for the hydrogens, half the glucose is present in that pyruvate, and there's 2 molecules of that was generated. And then that pyruvate gets fed into the mitochondria. CO2 is released, and the acetyl coenzyme A synthase then attaches what is left to that coenzyme A. And there you see it towards the bottom, CH3 double bond O attached to the coenzyme A. So again, all the energy that was originated in the sugar that we want to extract the energy from, we already generated 2 molecules of ATP in glycolysis, but the rest of the energy is then held in that CH3 double bond O, etc. And that acetyl coenzyme A now gets fed into the citric acid cycle in the mitochondria. Again, those are 9 reactions that we'll leave for biochemistry. But in the end, what comes out is hydrogens, H+, protons, electrons, carbon dioxide, and the coenzyme A, which is now free to be reused. Now, these protons and electrons just don't float around, but they're actually then become attached to a molecule of NAD. That's shown in the next slide. So this illustrates, so as a result of the TCA cycle, the citric acid cycle, you get the protons and electrons that have come off. Like I said, they don't float around freely, but they're attached to NAD plus by an enzyme called glyceraldehyde 3 dehydrogenase. It takes the protons and the electrons and adds it to NAD, which is called nicotinamide adenine dinucleotide. Again, another nucleotide, basically. And that NAD takes the protons and electrons, and that's converted into NADH. So So the electrons and protons are never freely floating. They just move into the NAD, make the NADH, and then that NADH then is fed into the electron transport chain. Once it hits the right appropriate region, then an enzyme called NADH dehydrogenase takes the protons and takes the electrons back off and then puts them into electron transport. And so in this next slide, this slide here, basically again just shows the outline of what we're talking about. So this just shows the mitochondria. So you have the pyruvate from glycolysis going in. You get the acetyl coenzyme A into the TCA citric acid cycle. CO2 is released, and then you get the electrons and protons coming off, and it goes into the NADH. And then that's what feeds the electrons and protons into electron transport. It's a process called oxidative phosphorylation. And so we're going to be looking at more detail in electron transport and the ATP synthesis. For me, this is kind of the more interesting part. It's all interesting, but this part here is really what I want to kind of focus on. And so let's look at what happens in electron transport. So this illustrates very simply what happens in electron transport. And we're going to look at more detail in the drawing that I made, which you'll see kind of, I think, describes things a little bit better. But you have the NADH giving off the protons, giving off the electrons. They get fed into the inner mitochondrial membrane, which carries electron transport. That's that line that's on an angle with the arrows, the blue arrows. And then you see the red arrow showing the pumping of protons in a particular direction. Electron transport is fed down this electron transport chain, released at the end, and it combines with oxygen to give off water. And so the products of cell respiration, just like the products of combustion, CO2 and H2O, except the energy is captured. So I want to look at a little bit more detail, adding my information to this kind of diagram. So this drawing represents, in a sense, electron transport chain in the inner mitochondrial membrane. And we'll look at the mitochondria later to get more of an idea of where that is. What you see up at the top and the left, there's NADH, and of course it releases the protons and the electrons by NADH dehydrogenase, and the electrons are fed into this electron transport chain. And there's around 15 electron carriers that carry the electrons from one carrier to the next. The way they do that is that the next electron carrier in the chain has a higher affinity for electrons, so one pulls the next, pulls from the one before it. Then the next one in line has a higher affinity, so it's able to pull the electrons from that previous carrier, and so on. So it carries in this very stepwise manner down this electron transport chain, and finally the final electron acceptor, once it leaves the electron transport chain, is oxygen to generate water. In fact, our breathing of oxygen is required for ATP production and electron transport. So there's around 15 electron carriers. They're all in the inner mitochondrial membrane. They are all clustered together in the inner mitochondrial membrane, and every mitochondria has thousands of these arranged in very specific orders along the inner mitochondrial membrane. We'll look at that later. In addition to the simple electron carriers, there are three larger complexes, which I have the names written out. There's NADH dehydrogenase, cytochrome BC1 complex, and cytochrome oxidase. These are large carriers. They're referred to as respiratory enzyme complexes. NADH dehydrogenase has 22 proteins. Cytochrome BC1 complex has 8. And cytochrome oxidase has 9 proteins in the complex, so they're very large. They are electron carriers. They transport electrons, participate in carrying electrons down the electron transport chain, but in addition, they function as proton pumps. And every time an electron or two electrons pass through these pumps, it activates a pumping mechanism that pumps the protons from one side of the membrane to the next. So when electrons pass through NADH dehydrogenases, 4 protons are pumped in one direction. Electrons pass through the cytochrome BC1 complex, 2 protons are pumped. Through cytochrome oxidase, 2 protons are pumped. And so as electrons are traveling down this thing, protons are being pumped to one side of the membrane. So this is a very unstable situation. This is what holds the potential energy. There's a high concentration of protons on one side of the membrane. The membrane is not permeable to protons. So there's a high concentration of protons on one side of the membrane. In addition, there's an electrical distribution as well, because the protons are positively charged. So you have both a chemical and electrical gradient on one side of the membrane. And so this represents potential energy. So how is this potential energy used? Well, it turns out in addition to the electron transport chain in the inner mitochondrial membrane, there are also numerous molecules, large complexes of a protein called F1 ATPase. Sometimes it's referred to as the proton pump, but it's also ATP synthase. This is a large multi-protein complex which in other cells uses ATP to produce a proton gradient, to pump protons. As it turns out, this is a reversible pump. It actually is referred to as a reversible machine, and I'll talk a little bit about that in a minute or two. But this is a pump or a machine that can be forced to work in the opposite direction. So in the presence of ATP, it hydrolyzes ATP, uses that energy to pump protons. But here it's used in a situation where there's an enormous proton gradient. And then, since the protons are so high in that gradient, the only way for them to get through the membrane is to pass back through this proton pump, force it to work in the opposite direction, and consequently produce ATP. It's actually a molecule that can be reversed in its operation. So what we have here is the generation of a proton gradient which drives ATP synthesis through the forcing of the protons through this complex in order to synthesize ATP. So this is what's referred to as oxidative phosphorylation. It's the phosphorylation of ADP to ATP. And with the production also of CO2. Our use of oxygen is required to generate ATP. The reason why we need oxygen is to produce ATP. So if you stop breathing, if somebody's choked, or if you stop breathing, the reason you die is because you run out of ATP very quickly. The oxygen is the final electron carrier and is used in order to generate ATP. In fact, the poison cyanide, sodium cyanide, can kill somebody basically in 10 to 20 seconds. And the reason why it kills people is it's a specific inhibitor of the cytochrome oxidase. So you inhibit cytochrome oxidase, it inhibits electron transport, it inhibits the production of ATP, and the person is dead in 20 seconds. So also what it shows is we use an enormous amount of ATP, and we constantly need it being produced. So let's look a little bit at this ATP synthase. So this is a diagram from the book, and this is a kind of a drawing of what we think the ATP synthase looks like. It has moving parts. The protons, which are shown at the gradient at the top, are forced through this pump, and in the process, it forces movement in what's called the ATPase head, and in that process the ATP is generated. Now this next slide shows the pump, which can work in both directions. So if you look at on the right hand side, it shows ATP hydrolysis. Like I said, normally in other cells, this particular protein is used to pump protons. It uses ATP as is shown. ATP gets converted into ADP plus phosphate, so that energy is used to drive the protons across the cell membrane. And because of the high concentration of protons as a result of electron transport, then it's forcing the pump to work in the opposite direction in order to provide ATP synthesis. So I find this process pretty remarkable. It's like the mitochondria has figured out a way to harness and take advantage of generating a proton gradient by taking advantage of normally a proton pump, but the proton pump can be forced to work in the reverse direction. So we've taken advantage of this pump that can go in either way, generating the proton gradient in order to make ATP. So this is actually, in a sense, a reversible machine. These kinds of proteins are referred to as really molecular motors or machines, and this particular machine can be worked in the opposite direction and get different results. And so people who are studying proteins, really they're beginning to look at proteins and large protein complexes in particular more as machines and motors that carry out work in the cell. And I want to show you just the first page of a paper that was published a couple of years ago looking at the F1 ATPase, the ATP synthase. And it's shown in the next slide. So this is a paper by people who were studying the structure and function of this ATP synthase. And what I want to do is just draw your attention to the language that people are using. That it's not the normal language of biology. It's more a language of engineering in a sense. So the title of the paper is F1 ATPase is a highly efficient molecular motor that rotates with discrete 120 degree steps. These are machines. Things like these, we'll talk more about them later. These are machines that have moving parts. They're machines that are built basically at the molecular level. I also want to read just a couple of other parts, just to give you the sense that the way people are thinking about these things and the way people are approaching molecular biology of the cell. So if you can enlarge the summary, or I can just read it. It says a single molecule of F1 ATPase, a portion of ATP synthase, is by itself a rotary motor, in which a central gamma subunit rotates against a surrounding cylinder made of alpha and beta subunits. Again, a completely different language, not what you might expect from biology. And then in the introduction, ATP, a major currency of energy in the biological world, is synthesized from ADP, and phosphate by extracting energy from the flow of protons across the membrane, which we just saw. This protein machine catalyzing this energy conversion process is the ATP synthase, which is ubiquitous from bacteria to animals to plants and animals. The ATP synthase is a reversible machine. It can pump protons in the reverse direction by hydrolyzing ATP. So it's remarkable. And the fact that the mitochondria takes advantage of that reversibility and makes the ATP that we need for all the functions of the cell. Now all this takes place in the mitochondria, so I want to spend a few minutes and take a look at what mitochondria look like and where these things are happening. So here's an image of the mitochondria. On the right-hand side, there's a electron micrograph cross-section through mitochondria. On the right-hand side is kind of a drawing. And there's a number of things I want to point out. there's a double membrane. There's an outer membrane. There is an inner membrane. The inner membrane is folded into what are called cristae, folded into numerous cristae. That's the inner membrane of the mitochondria. And the inner cristae is where the electron transport chains occur, where the pumps are and where the ATP synthase is. And the idea of having such a complex folding and extension of the inner membrane of the mitochondria is to increase the surface area so you can have many electron transport chains within one mitochondria. And cells that require more energy, like heart, for example, or brain cells, which require more energy, have many mitochondria. Some cells have very few mitochondria if they don't need a lot of energy, like skin cells. But cells that need a lot of energy, a lot of ATP, will have many, many more mitochondria, and the mitochondria will have more cristae in order to generate as much ATP as possible. And then there is the intermembrane space. And we'll look at that in a minute. That's where the proton gradient is generated. And then there's the matrix, which is basically the interior of the mitochondria, and that contains all the enzymes and what are required for the TCA cycle, which generates, of course, the protons and the electrons. Let's take a look at the next one. It's a little bit more illustrative. So this shows a simple, basically, diagram of mitochondria. So again, you have the outer mitochondrial membrane, you have the inner mitochondrial membrane with the cristae showing the folds. Oxygen is allowed to diffuse in. That's the oxygen we breathe. And that, of course, is the final electron acceptor for electron transport. But what you see is the electron transport chain shown in those three kind of greenish rectangles. Those are showing the pumps, showing the protons being pumped out into the inner membrane space. So you have the TCA cycle. It shows you the pyruvate coming in, the ethyl coenzyme A, the TCA cycle, generates the NADH, and that feeds that into electron transport. And then showing the pumping also shows the ATP synthase. And so the proton gradient is pumped in such a way that the intermembrane space is where the high concentration of protons exists. And those high concentrations is forced back through the pump. ATP is generated, and then the ATP is allowed to diffuse out. So the electron transport chains occur in the inner mitochondrial membrane. So this is my drawing of mitochondria, basically showing a similar thing. The outer mitochondrial membrane, the inner mitochondrial membrane, the cristae, the intermembrane space, showing the NADH, which is generated in the matrix, feeds the protons and electrons into electron transport chain in the inner mitochondrial membrane. Protons are pumped, generate the gradient in the intermembrane space, and then enforced through the ATP synthase to make ATP, and then, of course, that ATP diffuses out. Now, what's shown in this drawing, in my drawing, which wasn't shown in the previous drawing, is the fact that there's DNA. Mitochondria have their own DNA. Not only do they have their own DNA, but they have their own ribosomes, and they have their own transfer RNAs, and they carry out their own protein synthesis. So in a sense, they have their own genetic system. Now, if you look at the ribosomal RNAs, and if you look at the transfer RNAs, they're very similar to certain species of bacteria. And the thought is that mitochondria, also it's the same is true for chloroplasts in plants. The thought is that mitochondria originated separately as bacteria and then infected or was able to somehow enter a primordial cell way back eons ago. And because of the fact that these bacteria were able to generate ATP through this electron transport, that the introduction of bacteria into this primitive cell created a selective advantage because these cells were now able to produce a lot of ATP, and it became advantageous to keep these bacteria. And so they set up kind of a symbiotic relationship with the bacteria and the cell, and what we see now as mitochondria is the remnant of that. But they clearly they still have their own DNA, they still make many of their own proteins, and they still have their own transfer RNAs. And as it turns out, the mitochondrial genome has around 16,000 base pairs. It's not very big, but it still has 16,000 base pairs. And in humans, it encodes 37 different genes. And all of these genes that are encoded by this DNA either contribute to... So the mitochondria are semi-autonomous. They can't grow on their own, that's for sure. They don't grow on their own, but they clearly have a semi-autonomous genetic system, separate from our nuclear genes. So they have their own genome. Like I said, 37 genes. They encode for 13 proteins, which are involved in electron transport or some other aspect of oxidative phosphorylation. They encode 22 of their own transfer RNAs and 2 of their own ribosomal RNAs. So the ribosomes and transfer RNAs are different than what you find in the cytoplasm of cells. Now, like any other DNA, there can be mutations, there can be defects, and there are defects in mitochondrial genes that give rise to human diseases. There's around 6 or 8 human diseases that are known. They're called mitochondrial diseases. These are due to mutations in mitochondrial DNA that have some effect on one of these genes or more of these genes, and so they end up having a defect in electron transport. So I've listed a couple of the diseases. Like I said, there's 6 or 8 are known, but the 2 that I've listed are, one is optic neuropathy, and the other is called cardiomyopathy. And these are, the optic nerve requires lots of energy. And so if you have defects in mitochondria and ATP production, then there is a slow death of the optic nerve. Also, heart cells, cardio, require lots of energy. And again, if there's a defect in a mitochondrial gene that has an effect on the ability to produce sufficient ATP, then you end up with cardiomyopathy or some death of some cardiac cells. So there are clearly diseases that are due to defects in some of these mitochondrial genes. Now their inheritance is interesting. The inheritance is what is considered maternal inheritance. That is, we can only inherit these mitochondrial diseases from our mothers. It's not sex-linked. It's not considered sex-linked. It has nothing to do with the X or Y chromosome. It's different. It's maternal inheritance, sometimes called mitochondrial inheritance. And the reason why it's maternal inheritance is because we inherit all the mitochondria that we have from the egg. So the egg cell, which is destined to be fertilized and then develop into the organism for us, the egg contains all the mitochondria that eventually replicate and then make up our entire body. The sperm, on the other hand, only has one mitochondria. The sperm is illustrated by that little vertical line that's coming out on the left-hand side. They're very tiny. The sperm has one mitochondria, and that mitochondria is used to generate the energy for the flagella to work in order for this sperm to swim to the egg. But once it reaches the egg, that mitochondria is left behind. It does not enter the cell. The only thing that enters the cell is the sperm nucleus, which then fuses with the egg nucleus to make the diploid and start the developmental process. So all of our mitochondria originate in the egg, and so we consider that maternal inheritance. You can only inherit these symptoms, these diseases, from the mother. I do want to say it gets a little bit more complicated than that, and that is these diseases can be very heterogeneous. Sometimes what they call mosaic. So a mother that has the syndrome, let's say she has cardiomyopathy, a mother that carries one of these diseases clearly has mitochondria that are defective. But there are many mitochondria in the cell, many mitochondria in the egg cell. Also, there's many copies of DNA, and not all of them may be defective. So some of her mitochondria may be defective and some may not. So if we look at that egg, and let's say we have 20, 30 mitochondria that have defective DNA. When that egg starts dividing, it depends on where those defective mitochondria go. And that is all random. So they may end up being partitioned in such a way that they don't end up in cardiac cells or optic nerve cells. They may end up in muscle, or they may end up in skin, they may end up in liver. It just depends on the position of where those defective mitochondria are and how the cells divide. So one individual, one mother who, let's say, has cardiomyopathy can end up having children that look completely healthy with no symptoms. Or she may have kids who have cardiomyopathy, some may have optic neuropathy. It just depends on where those defective mitochondria end up. So it's variable in each family. And it depends on how many are defective and where those end up. Okay, so I'm going to end here. This is basically what I wanted to say about electron transport, oxidative phosphorylation. The main thing is how those electrons get to the electron transport, the function of the gradient, how that gradient is generated, where it's generated, how the ATP synthase is a reversible machine. These are the things that interest me most. And of course, structure of the mitochondria, which is very fascinating and has medical consequences. So in any case, I'm going to stop here. I'm looking at the timer. It's 48 minutes. I'm not going to, probably throughout the class, I'm not going to lecture for the entire hour and 10 minutes or however long the class is supposed to be. I think it's very hard to listen to a computer for that long. So as long as the lectures go, that's where I'm going to end the class. If a particular topic ends after 15 minutes, we're not going to stop there. I'll start another topic. But I'm not going to necessarily go the whole hour and 5 minutes or hour and 10 minutes, whatever the class is supposed to be. So we'll end here. Next time,